Good day, this is Professor Will, CBMD PhD. Today's Friday, September 25th, 2020. It is 10:35 and 48 seconds p.m. Eastern Standard Time, and this is a continuation of my microbiological research, particularly transcription. So let's hop to it here. Transcription presents a similar evolutionary picture, but with more and more pronounced exceptions to universality. The two largest catalytic subunits of the DNA-dependent RNA polymerase, B and B transpose, and eubacterial nomenclature are clearly universal, and show the canonical pattern, with again eukaryotic sequences being of the archaeal genera. But that is about it for homology. For the remaining main subunit of the eubacterial RNA polymerase, alpha, homology between the eubacterial and archaeal versions is only partial. Eubacterial alpha occurs in 2 copies in the holoenzyme, whereas its archaeal counterpart, found also in eukaryotes, comprises 2 separate proteins of very different size, each present in single copy in the holoenzyme, which parts of 2 showing homology to somewhat different parts of eubacterial alpha. The archaeal transcription polymerase has a number of additional smaller subunits, all of which occur in the eukaryotic enzymes, but not in the eubacterial. And as in the case of translation, the eukaryotic versions of the transcription polymerase contain a few additional subunits specific to themselves. When it comes to transcription initiation, one sees no homology between the eubacteria and archaea. The 2 mechanisms are different and use different componentry. Once again, the eukaryotes possesses an embellished archaeal version. Genome replication is an extreme example of archaeal and eubacterial difference. The closely related archaeal and eukaryotic systems are, to a first approximation, totally distinct from their eubacterial counterpart. And, as now might be expected, the mechanism of initiation of chromosome replication in eubacteria is fundamentally different from that basically common in the archaea and eukaryotes. In the past, it was common to draw phylogenetic significance, i.e. specific relationships from the fact that archaea and eubacteria both have circular chromosomes, in distinction to eukaryotic chromosomes which are linear. In light of the similarity between the archaea and eukaryotes in both chromosome replication and the initiation thereof, that conclusion needs revisiting. It is also of interest that both the eukaryotic and euarchaeal chromosomes show nucleosome organization, the single archaeal histone responsible being a homologue of 4 related histones that structure the eukaryotic nucleosome. This is an example of a theme that repeatedly occurs in the chromosome replication mechanism and elsewhere, namely a job is done by a particular multimeric protein complex. The many subunits therein, however, represent only one or a few gene families. In the eukaryotic case, a number of different members of a family tend to be represented in the complex, each present in single copy. In the archaeal case, there tends to be only one member of the corresponding family, and that present in multiple copies. Comparisons between the information processing systems of archaea and eukarya are what give rise to the strong impression of fundamental relationship between the 2. However, that specific relationship is reflected in relatively few molecules other than those involved in information processing, one example being HSP60. Overall, the relationships among the 3 basic cell types present in the complex, fascinating conundrum, a delightful challenge. Good day, this is Professor Will, CBMD PhD on microbiology, bioinformatics, and computer science. Well, hope you enjoyed listening. Until next time, have a great day.